Okay, so we're going to finish up carbohydrates today. And we're just going to talk about disaccharides. So we talked about formation of glycosidic bonds before, and now all we're doing to make these glycosidic bonds is linking another sugar with the first one. So if we identify the anomeric carbon down here, that is going to be the one that is involved in the glycosidic bond. And then what we can do is we can pick actually really any of these alcohols on the sugar to form a glycosidic bond. In this example here, we're just going to use the red OH, and we're going to react it with the sugar on the left at the anomeric carbon. And form a glycosidic bond. Now we need to have talk about a little bit more vocabulary, and that is how we classify these glycosidic bonds. And we classify them based on the orientation at that anomeric carbon. So this anomeric carbon right here, it is a alpha anomer, and we're linking carbon 1 at that anomeric carbon there to carbon 1, 2, 3, 4. So in this case, we would refer to this as an alpha 1, 4 glycosidic linkage or glycosidic bond. And then one other thing is this is also classified as a reducing sugar, and it's classified as a reducing sugar because at this end, it still has a anomeric or a hemiacetal at the anomeric carbon. So this can still act as a reducing sugar. In fact, this end on the right is referred to as the reducing end. Okay, let's look at a few more of these. So a disaccharide is two sugars hooked together through a glycosidic bond. And as we mentioned before, we can classify that bond based on the type of linkage. And so there's essentially three parts to this linkage. The first part being what is the configuration at the anomeric carbon. So that's where the alpha comes from. Second part is, well, what carbon are we linking to from the first carbon? Essentially, the one that we're linking the anomeric carbon from. And so in this case, it is carbon 1. And then finally, what oxygen are we linking to on the second carbon? And we're linking to the oxygen off the fourth carbon. And so that's an alpha 1,4 linkage. If we look at this case over here, this is an example of a beta linkage. So at the anomeric carbon here, this is on the same side. So that is the beta anomer, and we're still linking from carbon 1 to carbon 4. So that's a beta 1,4 disaccharide. Now these acetal linkages, just like we saw back in the ketone and aldehyde chapter, can undergo hydrolysis to release them into their individual sugars. And so if you just added water here, you can actually convert them into their individual components, where monosaccharides aren't converted into smaller sugars. So these disaccharides and oligosaccharides and polysaccharides that we've talked about before, the key there is that they can all be converted back into individual sugars. So we're going to talk about some common sugars, such as sucrose, which is actually a disaccharide formed from two monosaccharides, glucose and fructose. This is actually an interesting one that we'll talk about why in a second. But if you take some acidic water or you eat table sugar, your body will break it down into one glucose unit and one fructose unit. Cellulose, if you take cellulose, it has many glucose units linked together, and you can hydrolyze it down into individual glucose units. So back to the original vocabulary when we talked about monosaccharides, disaccharides, oligosaccharides, and polysaccharides. So disaccharides, oligosaccharides, and polysaccharides can all be broken down into their individual monosaccharides by hydrolysis of this glycosidic bond right there. So let's look at some interesting disaccharides. You may have heard of maltose before. You may have heard of lactose, which is milk sugar, and we just talked about sucrose, which is table sugar. So if we look at maltose, it is actually a linkage between two glucose units. So if we took the alpha anomer of one glucose and linked it to the 4 position on another glucose, we have an alpha 1,4 glycosidic bond. And so this is known as maltose, a disaccharide. Notice out here, we still have an anomeric carbon and a hemiacetal. And so again, that is a reducing sugar because that can open up and get oxidized. So maltose is a linkage that has an alpha 1,4 configuration. And this is actually a linkage that humans can metabolize. So it's actually an enzyme catalyzed degradation of starch. So starch is a polysaccharide that humans and other animals can digest. And so if you took starch and you broke it down into its disaccharide, you would have maltose. Now, if we take lactose, this is actually a linkage between galactose and glucose. And it's a different type of linkage. Instead of the alpha 1,4, it is the beta 1,4 linkage. But it's a beta 1,4 linkage between galactose and glucose. And remember, the difference between galactose and glucose is galactose actually at position 4 has an OH up, and glucose at position 4 has the OH down. So a little bit more about the anatomy of a glycosidic bond. This OH from the sugar on the right is always going to be down because that is glucose. This configuration right here is fixed by this sugar. The configuration here can change because that's our anomeric carbon. And so again, when we classify this, we tend to go from the anomeric carbon to the fixed carbon when we're numbering. And so again, we classify this as a beta, and it's at carbon 1 to carbon 4. So that is lactose, known as milk sugar. Now, some people actually don't have the enzyme or very little of the enzyme to actually break down that linkage. And so what happens when they drink milk or other dairy products, it tends to ferment in their stomach, and those people are known as having lactose intolerant or being lactose intolerant. Okay, here is another linkage. So this one is a beta 1,4 linkage like lactose, but here we're linking 2 glucose units together. And so this is actually the disaccharide if you took cellulose and broke it down to just 2 carbons. So let's review these. Maltose is a alpha 1,4 linkage. This is the linkage that starch has. Lactose has a beta 1,4 linkage, but again, it's between galactose and glucose. And then cellobiose has a beta 1,4 linkage, but it's between 2 glucoses. Now, this is a linkage that humans and most animals cannot metabolize. Even cows can't metabolize this. So when they eat grass, it's not the cows metabolizing it, it's the bacteria in their stomach. Termites can't metabolize it either. And so it's, again, bacteria in their stomach that actually break down the beta-1,4 linkage. Now, sucrose is a really interesting sugar. As I said, when we go to classify it, we tend to find the anomeric carbon, and we're going to figure out the linkage. And so there's one anomeric carbon, but what's interesting is we're actually linking to another anomeric carbon. So this is glucose on top, and we're linking from carbon 1, and this is the alpha anomer. So if you look at this, this is a fairly complex linkage because we're going from the alpha anomer at position 1 of glucose and if we look, we're going to this position here. That's the anomer. I know it's the anomer because it is sharing, that carbon is sharing 2 bonds with oxygen. So that is the anomeric carbon right there. And since this is a furanose made from a keto sugar, it actually has the CH2OH off of that anomeric carbon right there. And if we look at this configuration here, this is a beta. And we're starting from carbon 2. So this is an alpha 1 beta 2 linkage. And it's beta 2 on the fructose. So it's not very common to have anomeric carbons linked to anomeric carbons, and sucrose is, in fact, the only example that we're going to look at here. And so this is actually a non-reducing sugar, and the reason it's a non-reducing sugar is because both anomeric carbons are linked together. There is no hemiacetal in this structure that could open up and get oxidized. Okay. So again, table sugar is pure sucrose, a disaccharide that you can actually hydrolyze under acidic conditions into fructose and glucose. Okay, it is not a reducing sugar. It will not undergo mutarotation because there is no hemiacetal there. So it's connected as an acetal by both anomeric carbons. And again, we can identify the anomeric carbon because it is the carbon that shares a bond with 2 oxygens. So there's the anomeric carbon of glucose. Here is the anomeric carbon of fructose. And those anomeric carbons are connected through a glycosidic bond. So again, sucrose is a really interesting example of a disaccharide and has this alpha 1 beta 2 glycosidic linkage. So now that we can link 2 sugars together, let's link multiple sugars together. And if we link multiple sugars together, we end up getting polysaccharides. And so if we take glucose and just link it together in very, very, very long chains, we would get a structure known as amylose. And in amylose, the linkage is always an alpha 1,4 glycosidic linkage. And this example here shown in the box is just a short example. And so these chains can go on for quite a ways, several hundred residues. Now, amylose is the unbranched chain. You could imagine with all these other OHs out here that you could form additional glycosidic bonds to all of these OHs. And so you could end up with very highly branched polysaccharides. But amylose is actually just a straight chain of alpha 1,4 glycosidic linkages. And with this alpha 1,4 glycosidic linkage, it actually causes starch to coil up and actually form a helix. And you've seen this before in your chemistry experiments where you'll go to identify iodine or you identify starch by mixing it with iodine. And what happens is iodine actually fits together perfectly in this coil of starch. And when it does, it actually gets another iodine in there and forms this I3 positive complex that actually has a very deep blue to almost black appearance. And it's because that iodine can just fit perfectly in that helix there. And that's due to this alpha 1,4 glycosidic linkage, causes starch to actually coil up. Now, there is a related structure to amylose, and that's amylopectin. And this is a sugar that actually has linkages off of it. So if we notice here, here's just a normal amylose that has our alpha 1,4 linkage. But every so often, off of carbon 6, you get a linkage to another amylose chain. And so this is an alpha 1,6 linkage. And so what it does is it actually branches there off the chain. And so you end up with kind of a very long chain that actually has branches. And so starch is actually composed of a linear component and actually a branched component. And we'll talk about why branching is important a little bit later. Oh, never mind, actually right now. So amylopectin and glycogen are very similar in that they have the same type of linkages. They both have this D-glucose linked by an alpha 1,4 glycosidic linkage. And that's what makes the main chain, is this alpha 1,4 linkage. They also contain, both starch and glycogen contain this alpha 1,6 glycosidic linkage, or branch points. Now in starch, you don't see those branches as frequently, maybe every 25 residues. And in glycogen, you see them much more frequently. And so the reason for this is if you needed to release the glucose for energy, in plants, you'd actually have several ends where the enzymes could come and start releasing the glucose. So these little Pac-Mans are just representing the enzymes that are going to start at the end and chew off the glucose. Now, if you just had a big long chain, you'd only be able to release the glucose from either end. So it'd be very slow if you needed a quick source of energy, if you needed the glucose released. So by branching, you actually can have multiple endings and release the glucose much more efficiently. And so in animals, we actually store it in a much higher branched form. And the reason for that is humans and other animals often need to release the glucose very quick. And so in our fight or flight response, what happens is we dump a bunch of glucose to get us really quick energy, and it comes from glycogen. And so by having more ends here, we can actually release glucose very, very quickly into our body. Again, if we only had a straight chain, you'd be chomping off the ends and you'd probably be dead before you got sufficient amount of glucose into your bloodstream. But by having it highly branched, we can actually dump a lot of glucose into our bloodstream very quickly. And plants actually don't need to release it as quickly as humans. They tend not to need to run away from their enemies. Okay, so cellulose and starch are very similar other than the linkage. So he said in starch, amylose, the linkage is an alpha 1,4. In plants, it's a beta 1,4. And so cellulose is actually the main component of wood and plant fibers. And so just by changing this linkage here, you actually change the conformation of the polysaccharide. So here in this top example, you can see amylose. It's an alpha 1,4 linkage, and we told you that that actually coils up into a helix. Well, with a beta-1,4 linkage, it's pretty much straight. Now, humans can metabolize this linkage, but we can't the linkage of cellulose. So bacteria do have the enzymes to break that down. So cows tend to eat grass, put it in a stomach, and then they essentially let the bacteria work on it. And then they'll bring it up from their stomach, chew on it a little bit more, put it in another stomach, and eventually the bacteria will release the glucose that they need. But it's actually the bacteria that are releasing it, and the cows actually don't have the enzymes. It's actually thought that our appendix is a vestigial organ that actually allowed us to digest grass at one time. And then when we went to meat eaters, we didn't need that organ anymore. Okay, so if we actually look at the structure of cellulose and starch, again, starch coils up into a helix where cellulose actually ends up being very linear. In fact, you can actually get a hydrogen bond between the oxygen in the ring and the other OH. And so these end up being just very long and straight. And so they end up being very good at forming fibers. Just some other examples, variations on a theme. So there is this derivative. These are called amino sugars. They actually have an amine off there instead of just all OHs. And this is also used as a structural component. If you have an acyl group off of there, it's known as chitin, ends up forming a very strong and protective shell around insects. And so they have exoskeletons, and it is formed up mainly of chitin. And when the insect needs to grow, they have to shed their exoskeleton and then develop this protective skeleton of chitin again on the outside. So these are the arthropods. So insects, lobsters, beetles, and spiders, they will all molt and lose their exoskeleton and then have to create a new one. And again, chitin is a very, very strong biopolymer. Okay, that is it for this. So key reactions here, we've talked about hemiacetal formation, how these sugars will fold up to form either a pyranose ring or a furanose ring. We've talked about the reactions that they'll undergo. We can acylate them, make esters. We can make ethers. We can actually form acetals. We can reduce them. We can oxidize them. And we can oxidize them at both ends. We can lengthen the chain and we can shorten the chain. And that is it. Just as a side note, carbohydrates are very important. If you've ever wondered the difference between type A, B, and O blood, it's simply the type of sugars that you have on your red blood cells. So all blood has this arrangement. You got the N-acetyl D-glucosamine linked to a D-galactose and an L-fucose. This is one of the few cases in our body where an L sugar is actually prominent. And then what makes type B blood is you actually have an additional D-galactose on there. And what makes a type A blood is you actually have N-acetyl D-galactosamine. So everyone has this group in red in common, and then type B has a different sugar than type A. And that's why when you need to get blood, you need to match types. And we'll leave it at that, and we'll start with nucleic acids in the next lecture.